Hello, everyone. Let's get started. My name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I have just a few bookkeeping items to go over. First, questions will be responded to at the end of the presentation. Please enter your questions in the Zoom Q&A box. The audience can prioritize the order of the questions by clicking on the thumbs up button. 2, closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu on the bottom of your screen. Instructions are also provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Dr. Funda Meric-Bernstam, who is the chair of the Department of Investigational Cancer Therapeutics, the Phase 1 program at MD Anderson Cancer Center. She's also the medical director of the Institute for Personalized Cancer Therapy and the Nellie B. Connolly Chair in Breast Cancer. Dr. Meric-Bernstam.
Thank you so much. It is my honor today to moderate this session, and I'd like to start off by giving a little bit of an introduction about Jeff Chang, who's our speaker today. Jeffrey Chang is a professor at the Jackson Laboratory for Genomic Medicine, UConn Health. He leads a computational lab focusing on sequencing and imaging approaches to understand tumor ecosystems. He studied chemistry and physics at Harvard College, received his PhD in physics from MIT, and then took a postdoc at UCSF, where he studied comparative genomics and gene regulation. He currently focuses on machine learning approaches to predict clinical outcomes from multiplex image data, intratumoral evolution, and uses patient-derived xenografts for cancer modeling and preclinical trials. He's the PI for the NCI PDXNet Data Commons and Coordination Center, where I had the opportunity to closely work with him. And he's also the co-PI for the NCI Pediatric In Vivo Testing Consortium Coordination Center. Thank you, Jeff. We're excited to hear your talk.
Great, thank you very much. I will share my screen now. Okay, is this working? Shall I go ahead?
Don't see slides.
Okay, sorry. Let me get that going. Okay, how about now?
Much better.
Okay, great. Thank you. Okay, well, thank you, Funda, for that nice introduction, and thank you to the organizers for the chance to share the work from my team as well as our PDXNet consortium with all of you. I'm giving this talk really as a representative for the PDXNet Moonshot Initiative, and so I'm going to summarize the work that we've done together. And this is part of a talk called Cancer Discovery and Preclinical Validation Using Patient-Derived Xenografts. So what are patient-derived xenografts? Well, for those of you who are unfamiliar, this is a model system in which tumors that have been resected from patients are fragmented and then placed into mice. They can then be passaged and grown out in larger numbers. And this is a system which is very suitable for basic science as well as preclinical testing. And this is really one of the premier systems for testing right before we get to a clinical trial. And the mission of the PDXNet, when it was conceived for the NCI Moonshot program, was that these PDXs would be used to identify tumor characteristics that predict responses to target agents, and very importantly, to increasingly facilitate clinical trials. So the PDXNet Moonshot program started in 2017. This is a map of our participating members. We have 6 U54 teams, and they've been generating hundreds of PDX models, many data sets, and preclinical treatment studies. We work closely with the NCI, and especially the NCI Patient-Derived Models Repository. They serve as a central biorepository for all the models that we generate. And after those are validated, that's where any external researchers who wish to use these PDXs, they would contact PDMR to get them. And my team is from the Jackson Laboratory. We work closely with Seven Bridges Genomics to run the Corning Center, and we provide computational biology, engineering, and organizational expertise. I want to make a special shout out to our project manager, that's Dennis Dean. He is actually an investigator in his own right at Seven Bridges Genomics, and we're very fortunate that he runs all the data engineering and also many of our organizational activities. So this is a little bit more about the structure of the PDXNet. So the trial centers are shown here on the left. We have 6 teams. One is a combined team from Huntsman Cancer Institute and Baylor College of Medicine. Another is from Washington University in St. Louis. A third team is from a combined group from Wistar and MD Anderson. We have a second team from MD Anderson also, another group from Baylor College of Medicine, and another from UC Davis. And these groups work on various cancer types. Generally, the delineation is by the cancers that they specialize in. So we have a number of kind of internal processes which help us run our projects. We have committees for clinical translation, which really gets towards the clinical trial generation, as well as internal organization activities such as data sharing, joint research pilots, and organizational decisions through our steering committee. Our key deliverable, aside from the clinical trials, are the PDX models that we're generating. We also provide a number of raw data sets, such as sequence data associated with the xenografts, as well as processed information such as gene expression, variant calls, and of course, the drug response data and other information such as histology imaging. We have a number of end users in mind. These are especially, again, as I mentioned, to generate clinical trials through the ETCTN network. We also have processes where we work with various NCI-funded investigators, other consortia, and also the Department of Energy. So the main outcomes that PDXNet has generated over the last few years since we've been in operation is, first, we've generated 10 CTEP letters of intent towards clinical trials. As of early 2021, we had 88 publications from original research projects. And we've also generated 690 new PDX models that have either been fully submitted or now in process. So about half of these are already fully submitted and being made available through PDMR. And the others are kind of going through these final stages. We also have a public data sharing portal, which is available to anyone in the community at portal.pdxnetwork.org. And I'll describe a lot of the features in that during this talk. In addition, we have 16 validated PDX data analysis workflows where we have done tasks which come up frequently and really paid a great deal of attention to the high fidelity and robustness of these tasks with our bioinformatics expertise. And then in addition, we've had a number of consortium-wide collaborations, and these include a multi-center reproducibility study for drug response, and that was published in Cancer Research. The second was on the genomic fidelity of PDXs during passaging, which has been published in Nature Genetics. And the third is a clinical target composition in these PDX collections, which has been published in Nature Communications. So this first study, which was really kind of the most important activity for the network, was to evaluate PDXs and as a system for preclinical studies. And one of the problems that the network had discussed at the beginning of our projects together was that many groups have been doing xenograph studies, but it's often difficult to tell if one group's results are believable. And that's because there are differences in how people raise their mice. There's differences in the process in which the tumors are engrafted, and the drug administration can also be quite different. And so it had been non-obvious whether the robustness of the results in PDX studies was sufficient to overcome those empirical differences in procedures. So what we did to address this issue was we did, for the first time, a study in which we blinded 4 different groups, and we gave them 3 different models, and we also told them a drug to try, which is temozolomide. And we said, tell us which model is responsive. And that was almost the complete specification of the project. We did not tell them anything about which model was likely to be susceptible. We did not tell them anything about the drug dosing or timing. We didn't tell them how many mice to use. We just simply said, here's the drug, and tell us your answer. And then we were very pleased to see that actually after doing these studies independently, all 4 groups came up with the same answers. And this is just data from 2 of the models. The progressive disease case, all 4 groups found that under both control and then the temozolomide treatment, they had progression. And then in the case of a model which we had expected there to be tumor shrinkage, this was also found identically by all 4 of the groups. So in fact, despite these variations in procedures, the PDX system was robust enough to handle the differences in how groups were running the experiments. So the second thing that we studied was the evolutionary behavior between the primary tumor and the PDX. As we know, when you take a sample of a tumor and then you place it in your environment, you'll only get a subset of the cells from the original tumor, and that is inherently an evolutionary process. So what we had been concerned about was the possibility that there might be some special selection for particular subclones that would have genomic differences from the primary tumor. So we engaged in a study in collaboration with the EuroPDX consortium, which is of similar size to the PDX-NET. And we looked at this question. First, what we're seeing here are trios from primary tumors, early stage PDXs, and late stage PDXs. That's these 3 rows. And what we compared was first colorectal cancers. The data on the left here are a summary of 87 colorectal cancer trios with the amplifications and deletions. And what you should see here is that there's very little difference between the primary tumor and either the early stage PDX or the late stage PDX. And that's also true for the breast cancer. So this is actually a summary of 390 genomic data sets. And this really clarified the point that there is relatively little tumor evolution in going to the PDX. In addition, we did some experiments where we compared how the primary tumor and the PDX varied versus a case where you would just take different multi-region samples of patient tumors. And what we also found was that there was very little difference in that range of variation in those 2 situations. So basically, the difference between the PDX and the primary is no greater than what the spatial variation is within patient tumors themselves. So those works were both published in the last couple of years. So now I'm going to move on to talk about some things we've been doing more recently. One of our emphases now that this first round of the moonshots is nearing its end has been to think about the data sharing. So we recently published the PDX-NET portal in Nucleic Acids Research Cancer. This just came out earlier this month. And this provides all the data that we have generated as part of the consortium to date. So within this, we have 334 of the models here so far. That is a moving number and rapidly accruing. And this work was led by 2 very talented scientists, Senator Koch and Mike Lloyd. So in addition to the raw data itself, we have these data analysis workflows. Currently, we have 16 data analysis workflows, which are available through the portal, but you can also run them directly on the Cancer Genomics Cloud, which is the platform that Seven Bridges runs as supported by NIH. So these are immediately runnable through an Amazon backend. And some of the tasks which we have enabled are expression calling, RNA fusion calling, CNV calling, mutation calling, et cetera. And in particular, all these pipelines have been very extensively validated by comparing to the processes that each of our consortium members have been using, as well as the processes that have been developed at the patient-derived models repository with NCI. So I think we have much more extensive robustness testing here versus what would be possible in a single center. And so these different pipelines have been extensively requested by academic and industry groups. And what we're seeing is that this is providing a significant benefit to the community. So the kind of sequencing data we now have about 2,800 files. These are of various types, including the exome-seq and RNA-seq data from the different contributors. This is 873 total actual biological samples from 307 different patients. We're also working with some of the main groups who are very involved in the PDX field. Specifically, we have been working very closely with the Patient-Derived Models Repository. So they have shared their data with us, and this is an even much larger set comprising about almost 15,000 sequence files and nearly 4,000 samples in about 600 patients. So one of the reasons for consolidating this data together is that Now, all the data can be analyzed in a common framework, which is really, I think, one of the great needs for facilitating the faster development of clinical trials so that we can take data mining approaches on these PDXs and analyze them in a systematic way. One of the new data types which we have been emphasizing recently has been imaging data. So we have been collecting the histological images from our partner groups. So we have more than 500 of these images so far. We also have a number that are still in process, about 500 that are on the way as well. And so this is going to rapidly accumulate. Some of you who are in the audience who are following the digital pathology field may know that this area is rapidly expanding. So we're consolidating this now kind of in preparation for joint analysis of histological and genomics data. And of course, the most important data type is the treatment response data. In order to further develop trials, what we want to make possible is for groups to mine this data. So currently we have 20 treatment studies which have been made fully available on the PDXNet portal. And this comprises 10,000 volume measurements at individual time points. So we have a lot of additional data in process. We're working with many of our partner institutes on this, as well as the patient-derived models repository. And we hope to increase this quite significantly very soon. In addition, we're now at the phase where many of the initial studies that were launched by the PDXNet members are at the publication stage. So those are rapidly accruing in terms of what we can put online. And as part of this, we are developing a manuscript, which will be a comparison of statistical approaches for evaluating PDX treatment response. And this is a project which is being led by the MD Anderson group and some biostats groups from NCI. And one of the things that we're kind of looking at for this project is, in addition to the actual formal statistical evaluation, we're also making available multiple visualization approaches on the portal page. What you see on the left here, for example, is a set of studies where we have treatment response as a function of time for various PDX models. And what you can see here is that we've normalized the response data on a scale of -100 to 100. So 100 is kind of the most extreme time point. Under various kind of parameters, we may, for example, say that 100 would be if you reach fourfold times the original tumor size, but -100 would correspond to complete regression. And we're particularly interested in that because from a clinical trials perspective, that is really what we're aiming for. We're looking for those cases of complete regression or stable disease. And this kind of formalism makes that more clear. So for those who are kind of looking at different sorts of aspects of the volume response, we're building some of those features into the website. So now I'm going to talk about the image analysis in a little bit more detail. And so this is a project which is being led by 3 of our scientists, Brian White, Xingyi Wu, and Center Coach. And so this is an example from the field of digital pathology. From my group, we published this earlier this year. So this is a project where we predicted HER2 copy number status from H&E images. This is on the left, an H&E image where the pathologist had labeled where the tumor was. And we knew ahead of time that this was a non-amplified HER2 tumor. And then when we put this into a convolutional neural network model, indeed, we were able to recapitulate that this was a HER2 negative tumor. As you can see, it's predicted correctly and in the right locations. So this idea is what we're trying to develop as part of PDXNet. We are in the process of establishing a robust image analysis pipeline. And this involves multiple steps. First is general QC, which we're using a package called HistoQC. And then we have stain normalization, and then various deep learning inference aspects. So this is a pipeline which uses an inception-based convolutional neural network to make calling of things like tumor normal or subtype status. We also splinter off into a cell segmentation calling, and for that we're using another neural network called HeverNet. And this is work which is in collaboration with a number of teams throughout the consortium and also the UT Southwestern group, including Shidan Wang, Yang Xie, and Andy Shao. This is an example of the kind of data that we've been looking at. This is a histology image from Baylor College of Medicine. On the left, you see a breast tumor where there's a clear delineation of the stromal regions, the tumor regions, and the necrotic regions. In fact, the BCM team has annotated the individual cells, which has enabled us to do comparisons of how these computational predictions work versus the ground truth. And what we're seeing is that these methods, which have been developed for clinical samples, still work quite well in PDXs, despite the change in the microenvironment, especially with the mouse stromal context, and especially for the tumor cells. So for the tumor cells, we can identify them 98% of the time, or at least segment them. Whether we call them definitively as tumor cells, that's a slightly more difficult task. And we're calling them as tumor cells about 3/4 of the time. And so this is a project which is being done by Xingyi Wu, John Landu, and Todd Sheridan. This is an example of what that looks like again. So this is from a LUSC xenograft. And Todd has annotated the necrotic stroma and tumor regions from an H&E slide, and that's what you see on the left. And these are predictions from this neural network HoverNet. And it's a little bit distorted here, but I hope what you'll see is that there is kind of a ring structure, which is very similar to that on the left side of the image. And that's indicating, again, that we can properly predict the necrotic stroma and tumor regions, even within this PDX context, which is not obvious that, that these trained methods would have worked for. This is a little bit more detail on that. We have a random forest-based method which can classify these regions quite accurately within PDX images. Okay, so that's really kind of a summary of what we're doing within the coordinating center. I now want to describe some of the exciting vignettes for individual research studies and letters of intent generation from our testing centers. So the first of these is from Baylor College of Medicine, and this is work from Mike Lewis's lab, and he's been studying triple-negative breast cancer. Especially what he's been looking at is combination therapy studies of chemotherapy. And particularly what he did was he took a panel of several dozen triple-negative breast cancers and treated them with carboplatin and docetaxel. So what you see on the top here are both single treatment studies and also the combination. And so the single treatments are in blue and green, and then the combination is in red. And what you should see here is that the red, the combination, generally is the best, but it's not obviously better than the best single treatment. And Mike has found this to be kind of a general rule, at least for these two therapies. And that's indicated here. What you see is that if I take the single best response out of carboplatin or docetaxel, it's not statistically significantly different than the combination therapy treatment. So Mike has then been exploring, why this is the case. And so what he's looked at is the gene expression patterns of the PDXs that respond to chemotherapy versus those that don't. And so this is a gene ontology analysis of that behavior. And there's a lot of biology here, which I won't go into great detail on. But what you can see is that, for example, folding and stress response seem to be a pathway that is higher in these resistant PDXs. So that's right here. So Mike has followed this up by targeting IRE1 in the unfolded protein stress response pathways, and found that if he targets that with an IRE1 inhibitor, in fact, he can overcome this chemotherapy resistance. So that's particularly obvious in this BCM3107 case. You can see that the response with just docetaxel or just the response with the IRE1 inhibitor is quite modest. But then when those are used in combination, there is a very strong quelling of the tumor. These other cases show a more limited effect, but the overall behavior is quite similar. So Mike has been following this up by doing a number of other combination treatment studies of targeted therapy with carboplatin. What you can see at the top of this graph are the single treatment studies for various targeted therapies as well as carboplatin. And in fact, what Mike has found is that there are a number of cases where the targeted therapy can enhance chemotherapy response. And that's highlighted at the bottom here, where you can see that the combination is doing better than the carbo for these squares with the purple ring around them. So these are very promising for further study and development of letters of intent. Mike is looking further into the mechanisms behind these as well. I'll next go to another vignette, which is from the UC Davis team. So the Davis team, led by Luis Carvajal Carmona, has been looking at cancer health disparities. And one of the things which I think many of us in the field know about is that the models that we have now, and also the kind of big sets like TCGA, they don't have a proportional distribution of ethnic groups. So this is shown on the left. This is the ethnicity distribution for TCGA. This is particularly for the group of Latino / Latina versus other groups. And then this is also for cell lines, which are also quite skewed. So this is an important problem because it may be that there are mechanisms and also treatment susceptibilities that are quite specific to particular ethnic groups. So they've been working on this problem. So in particular, they've been focusing on gastric cancer. And so this is just a schematic showing the different subtypes of gastric cancer. This is from the original TCGA gastric cancer paper. And so Luis is looking at questions such as, if he focuses on these minority populations, is there differential responsiveness to treatments for gastric cancer versus non-Hispanic whites, looking at driver alteration effects and then risk factors. So this is an illustration, again, of this. So on the right here, this is the TCGA gastric cancer group and the ethnicity distribution, and again, broken down by Latino versus other. And then this is the group that is from the UC Davis team. So UCAMP is their project group. And in their group, they've managed to obtain PDXs, which are especially containing these minority populations. In particular, here they have a lot of the Hispanic Latinos, that's shown in the teal color here. And interestingly, if you look at the TCGA-defined molecular subtyping for these two groups, what you see on the top here is the distribution of subtypes for the UC Davis group. So in particular, notice that there's a lot of the genomically stable subtype here, but in the TCGA set, it's much smaller. So there does seem to be some kind of difference in the subtyping as a function of ethnicity. So to address this problem, they are developing a large number of new PDXs and also patient-derived organoid models, and are going to get up to 200 of these. And then they're going to progress to various preclinical studies to develop letters of intent as well. As part of this, one of the computational tasks which we have been working on together with Luis's team is to generate a better estimator of genetic ancestry. One of the challenges for this field has been that there is not good reference data for Native American populations. And this is not only a challenge from the reference group, but also because these populations are highly admixed, the computational challenge is quite non-trivial. So we are in the middle of implementing a pipeline which can handle these Native American populations with high fidelity. Another vignette I'd like to talk about is from the melanoma MD Anderson group. And this is a project led by Vash Yanunanda and also Michael Davies. So in melanoma, really the key mutation which people have been looking at for years has been in the V600 mutation in BRAF. Half of cutaneous melanomas have this mutation, and the standard treatment has been BRAF inhibition with a MEK inhibitor. And so you get about 15% complete responses for these cases, but 90% of them still have recurrence. So the key problem for that group has been to develop ways to overcome de novo resistance and also acquired resistance. And so the strategy which they have been looking at has been targeting an additional gene, BCL2, along with BRAF and MEK at the same time to prevent de novo and acquired resistance. So this is some of their initial studies from that. So what they saw was that if you look at BCL2 itself, based on IHC, they found that if you had a high level of BCL2, then you had a very rapid time to progression. And lower levels of BCL2, then you would have slower progression. They also observed that after treatment with the dabrafenib trametinib combination for the BRAF and MEK inhibition, you would often have an increase of the BCL2 IHC. So what they decided to do was target BCL2 with an inhibitor, and they've been doing this in PDX studies. And this has been in collaboration with another of the PDXNet teams at the Wistar Institute. And so that is summarized here. I will skip to the results. And so what they've been particularly looking at are these markers of potential response to the BCL2 inhibitor, specifically the BCL2 status, but also some associated markers, especially MCL1 and MITF. And so based on those markers, they predicted that certain PDX models would be sensitive and certain models would be resistant. And in fact, their predictions were quite accurate. They saw that this group predicted to be sensitive, had a very good response to the triple combination, including the BCL2 inhibitor. They've tested both navitoclax and venetoclax. And this group that was predicted to be resistant showed a much poorer response. So in summary, these results were quite promising. They've also done some additional stratification based on the behavior of MCL1 itself. So to follow that up, they have been doing other experiments with MCL1 inhibitors. And so what you see here is that based on the basal MCL1 level, that seemed to have an impact on PDX growth inhibition. And they saw that if there was high basal MCL1, you would in general have a poor response to the triple combination of the BRAF, MEK, and BCL2 inhibition. But very excitingly, what they saw in a tumor where there was this MCL1, that was very high. When they did a combination of BRAF, MEK, and an MCL1 inhibitor, they completely regressed this tumor. And in fact, at the time of harvest, there was no visible tumor. So that was very promising. And this MCL1 inhibitor seems to be a potentially significant target, because if you look in TCGA, a very significant fraction of tumors have MCL1 amplification. So to summarize their work, they've seen a very interesting and unexpected efficacy of the combination of dabrafenib and trametinib with the navitoclax or venetoclax. They're stratifying by these biomarkers, BCL2 and MCL1, and they have been working now with Ascentage to get the BCL2 inhibitor pulsitoclax through CTEP. They are working now in a phase 2 trial to evaluate this DT combination with navitoclax. And then now they're doing some additional studies on the MCL1 inhibition to consider these other sort of subdivisions of the behavior. I want to now move on to another vignette from the WashU team. One thing that WashU has done, and this is led by Li Ding, is to analyze the mutation and the expression data within the models which are being generated by the PDXNet teams and also PDMR. And so this is really kind of the main activity that I think most external groups would be interested in, which is what is the likely susceptibility of these various PDXNet models to different treatments. And so they've essentially cataloged that, and what you see are the various mutations. So I won't go into great detail on this, except to encourage those in the audience to look at this paper if you're interested in using any of the PDXNet or PDMR models. A couple of things that the WashU team has done have been to generate letters of intent, specifically Cynthia Ma's team within WashU has developed an LOI for eribulin and capanlisib in metastatic triple-negative breast cancer. Kian Lim's team has developed an LOI for a MEK inhibitor and the DS8201 therapy in metastatic pancreatic cancer. In the interest of time, I will describe these a little bit more briefly. So what they've seen, for example, is that their TNBC models are sensitive to eribulin in some cases and other cases not. They've also seen that there is an effect with capanlisib. I'll just kind of describe the trial rather than describing all the basic science. What they've done is start a trial in August 2020, and it's having ongoing accrual at 6 ETCTN sites. For the work with Kian Lin's team, they've been looking at this pancreatic adenocarcinoma and other KRAS-mutant solid tumors. This is this very interesting case of potential HER2 targetability by the DS8201 ADC, and also the relative impact of RAS mutations. And finally, I want to talk a little bit about some work from the Anderson team. One of the studies which they did in the early stages of the PDX-NET was to look at cyclin E overexpression and how it may sensitize triple-negative breast cancer tumors to WEE1 kinase inhibition. And so this is some of their early data showing that for PDX that did not have cyclin E expression, there was very little effect of this WEE1 kinase inhibitor. But for these other two PDX models, which had the cyclin E, there was a stronger response. And so based on this, they developed some clinical studies really across a number of tumor types. I think some of their exciting results are that they found within patients, 7 confirmed partial response with an overall response rate of 25%, especially in high-grade serous ovarian cancer. They found very interesting results with 38% of the patients having partial response and 8 with stable disease after 6 months. And then finally, I just want to mention, what is the big challenge of these xenograft studies? It's their speed. It's hard to generate these experiments as fast as we would like, and that is a limiting factor to generate further clinical trials. So one of the members of our consortium, a team led by Ilana Whelm, together with Brian Whelm, they have been working to develop a platform for combined xenograft and organoid use to increase the speed of discovery. So what they do is they generate organoids from the xenografts, which gives them a steady supply of these organoids. And what they found is that within their models, they see a very high fidelity between the drug response in the xenografts and organoids. So this is an approach which we're very optimistic can allow us to increase the speed of getting treatment response results. So let me now conclude and summarize. So xenografts are a robust model system which is genetically similar to patient tumors and have a reproducible treatment response. We're seeing that within the PDX-NET, we're generating clinical trials across diverse cancer types. And now that we're in this stage where we want to increase the speed of these, we're really focusing on the data sharing. So we provided a data portal that has genomics, histology, and treatment response data. We also have validated pipelines that anyone in the community can access. Again, we have very well-validated sequence analysis pipelines with the tumor volume and image analysis pipelines in process. And these are all helping us towards the goal of facilitating target discovery and treatment studies to further accelerate clinical trial development. So I'll finish there, and these are the team members. I've highlighted the main PIs who are part of the PDX-NET consortium. These are the key members from my team together with Seven Bridges, who are in the coordinating center. And the work that I showed on the fidelity of PDXs and patient tumors is together with the EuroPDX consortium. And we certainly have many, many more consortium members and collaborators that I was not able to show. So I'll stop and thank you very much for your time.
Thank you, Jeff, for that great presentation. This has been a lot of work across a lot of teams, and really, you guys did an amazing job coordinating it. So I think the success of the group working as a team is at least in part attributable to the coordination of the center. I see that we already have some questions coming in for your interesting talk. The first question is about the image analysis pipeline. So I'll just read. The imaging analysis pipeline is very exciting, especially the incorporation of deep learning approaches. Given the breadth of sample types across PDX-NET, could you describe how PDX-NET decided on which data sets for which images to use for the training models?
So the first, I think the practical question is the availability. So we worked with what we had, certainly from Anderson, they had some nice annotated data sets that Funda helped us out with, as well as Jack Roth. and Bingliang-Fung. So we've been especially focusing on lung adenocarcinoma and lung squamous cell, but PDXNet particularly has a lot of breast cancer samples. So we've looked at that as well. We have a big set of images from PDMR. Again, a lot of breast, but not only. So we're focusing on that. And in addition, we have some others from JAX and a number of other groups. I think in total, we're going to end up, at least for our establishment of the deep learning pipelines, we'll have more than 1,000 images, including about 100 cases where we have matched primary tumor and the PDX, where we'll be able to investigate robustness issues. So I wish I had a bit better logic to it, but it's really the sample availability, because many of these images were collected before people had in mind that we could do this deep learning based analysis.
Thank you for that answer. Just to sort of do a little bit more of a deep dive, what are the differences, do you think, in sort of human sample analysis versus PDX analysis? You mentioned some of the more complexities due to the different stroma.
Yeah, I think that will be the main thing. What we've seen is that we can separate things at this broad level, which is tumor versus stroma writ large. But certainly, if we have tumors that are prone to high levels of immune infiltration in the clinical context, then I think those will be more challenging. So I think that's what we need to explore. There is certainly the possibility that some of the fine scale immune processes will still be detectable in the PDXs. For example, I think many here probably know that if you look at the gene expression data, you can still see signs of things like antigen presenting cells. And it's possible we'll see those in the histology as well. But in terms of, I guess, the more canonical immune cell populations like active T cells, I think that's where we're going to be, it will be different, and we have to figure out what's possible.
Funda, one question drifted into the chat box if you want to try to find it.
Yeah, I saw that actually it was a comment about wanting to collaborate. So Jeff, you have people that want to collaborate with you. I don't see a question per se. So that's great. That I'd like to remind everybody that, if you have any questions, please put in the question and answer.
Jeff, has PDXNet got into the so-called co-clinical trials or avatar kind of models?
I think the Funda actually knows the most about that. It has not been our main line of inquiry, but she is doing something of that direction. Maybe Funda, you can take that.
Yeah, thanks. So as you know, PDXs are used in a variety of ways, and actually some of the sort of early PDX development papers really did focus on the patient treatment selection angle. We've spent a lot of time discussing whether that's something we can go into, just as a, not as a PDX endeavor as part of PDXNet, but as a general principle. I have to say that there's, especially for advanced disease where we really need novel therapies, the timeline to be able to generate a PDX and test it is sometimes too long to benefit a patient. So it's not a not doable thing, but more complex. So one area that we've discussed a lot is whether we can build a, molecular profile and, PDXs like my tumor kind of an endeavor. And that's, I'm hoping, something that we can work together as a community to build so that we can immediately identify molecular matches and to be able to sort of predict therapeutic sensitivity. We definitely have had a lot of precision oncology based on genomics alone, and it's quite clear that that only scratches the surface. So we need sort of collaborative team efforts that involve all scientists across the world.
I see another question drifted into the chat box, Funda.
I see a question in the question and answer. Using a novel 3D tumor organoid model that better mimics human tumors and that allows for establishing the inflammatory environment. I actually don't see the full question, but maybe you can give a little bit more information on the 3D organoids and advantages, Jeff?
Yeah, I can talk about, certainly this is a key issue for the field. So far, we've had a lot of teams that are interested in making humanized models with the PDXs. It's for PDXNet formally, that's not within our kind of our focus, but the teams are very interested. As for organoids themselves, the system I was describing from the Whelm group, that is not humanized. But I think our members are interested. So I think the short answer is that we don't have that yet. I certainly believe it's important, though.
Another question was whether PDX models will be required for NCI funding on specific type of cancers, and if so, which ones will this change happen in?
I don't think I can really answer that question. That's for the program officers. But I think from a basic science perspective, the challenge has been, we know that cell lines are useful, but maybe not quite as close to the original tumor as we would like in many cases. So it's really a question of the tissue context. And certainly, I would expect that things like cancer stem cell-like behaviors would be better modeled in a PDX than in certain cellular pure cell line model contexts. So I think that's really a challenge for all of us in the field. And my own personal hunch is that as we generate more of the spatial transcriptomics data, we'll get a better understanding of which cases that is relevant or which it is not. Certain tumors, they're quite uniform internally, but in other cases, the spatial structure, not only for the stromal interaction, but also the tumor itself is rich.
A question came in asking about the minority PDTCs and the overall PDX network addressing health disparities.
So for that topic, what we've been able to do so far is generate new PDX models. So I think that in this initial stage that, generating a PDX model itself is, if things go very smoothly, it will take a year. But often they don't go that easily, especially if you're dealing with models which are from a very slow growing tumor type. So we're at the stage of generating more models, and we have several dozen models which are in process of that nature. And I think in this, the next iteration of the PDXNet, there will be a lot more studies which will now be enabled for that. I wish I could say we had made further progress, but really it's at the model generation stage.
Another question about model availability. I see in the portal that there is a limited number of neurological PDX models. Is that due to the complexity of creating them or due to the focus of the groups in PDXNet?
It's more the focus of the groups. There are PDX models from other teams that are neurological. For example, within this pediatric cancer consortium, which I happen to be a part of, there are a number of teams looking at neurological models. The PDMR does have some of those. And I would say if you're looking for those, you can also look at some of these broader resources for finding PDX models. PDX Finder is another consortium which is funded by the NIH. So I think that's a good resource for you.
Yeah, aligned with that, a question was, how do you help people learn how to use the workflows and other resources in PDXNet portal? Are there webinars, trainings, courses, etc.?
Yes. So we get requests for those very frequently, and that's what our colleagues at Seven Bridges, especially led by Dennis Steen, that's what they do. They have a team of three analysts who contribute to that. So I would say just look at the website, the PDXNet portal website, and then we have contact information there, and just send us an email if you still have questions. But we found it works pretty well because everything we have is based on this containerized model within the Amazon cloud. And the idea is this push button genomics where you upload your data. So it's a little easier in general than if we had to work with you on your high performance computing system.
Another question about the genomic evolution data that you presented. The exact question is, I noticed you have very systematic comparison of patient tumors and PDXs at copy number variation level. Did you ever compare the somatic mutations in early PDX and late PDX, or in different ethnicity like Latino background PDX with different patterns versus Caucasian?
Right. We didn't do that, I think, in the way that you're asking for. And one of the challenges of that is it it's not it can be difficult to have a good null model for the rate of mutational evolution, especially with single somatic mutations. And it's also particularly complicated by cases when you have these very fast mutating tumors, especially, for example, if you have TP53 mutation, you'll just get more after that. So we didn't do that. It's an open question if you're interested in studying it.
Yeah. I do want to add, though, the copy number change data was in part, triggered by a paper that suggested there was a lot more genomic evolution, and was partly why that was a focus. Back to the disparities question, for the minority PDTCs, could you comment on how your team was able to build trust with these special populations to acquire these very precious samples?
Yeah, that is absolutely critical. And I really want to compliment the team from UC Davis, especially, who has worked on that. The PI, Professor Carvajal Cremona, I think it probably helps that he is from a Latino background, and he knows these populations well. And so they have just put in a tremendous amount of effort to build rapport. I think it's there's no easy answer to that except to get to know the people that you're working with and make sure that that they are happy with the approaches that you're going to take with it. And also one of our other teams from Baylor College of Medicine, they have been focusing on particularly the African American populations, and that's they're in the Houston area, and so the groups that they're getting from are especially in this urban setting where they have a high level of diversity. So that's been kind of a different dynamic, but certainly a great effort to make sure that that happens as well.
Okay. I think you've handled all the questions. Great. More questions. Does PDXNet work with other cancer moonshot programs using imaging approaches such as H10?
We don't have any formal relationship, but I think that those things will become easier. So some of our consortium members have projects that are part of HTAN, but we don't have anything that has been established between the consortia. I think that will become part of our normal process, though, because really the imaging work that we're doing so far has been to prepare for this kind of period where I think everyone can see the technology is rapidly developing. So we want to make sure that those types of collaborations can be possible very quickly.
Great.
Funga, one popped in the chat box. If you don't mind going over there.
Yeah, are they recording the country of origin of each Latino Hispanic? There's a lot of variation.
I don't know the answer to that. I wish I could tell you. What we are looking at, I think the key analysis we're doing there is the admixture analysis, and with the populations that we've kind of brought in. I wish Luis were available to answer this question, because I'm not exactly sure how they obtained their Native American populations. What we can see is that there's a pretty clear kind of linear admixture separation between the European Caucasian populations and the Native American. What we don't see is some kind of like diverse phylogenetic tree. So it's at least simple enough that we can do that.
In order to study tumor organoid and treatment response, what imaging technique would you recommend? That's a great question, actually. I'd expand it to say which assays.
For organoids, that's a really interesting question. Outside of PDXNet, my team works on this a little bit. And I think, for example, what we're doing in this PDXNet independent project is we're labeling individual cells with fluorescent markers, and we're mixing different kinds of tumor cell populations, and then we're trying to see which cells will respond to treatment and which will not. And we're also doing this imaging in 3D. So our technology for that is confocal microscopy in using a machine called Opera Phoenix from PerkinElmer. So I think there's actually a lot of room to discover things even from just fluorescence-based. If you can get, or DAPI even is quite informative. But there are certainly things I think that would be maybe specific to the type of tumor interaction you're looking at. For example, if you are looking at an immune cell interaction with the tumor where you're doing a co-culture and you're interested in something like a dendritic interaction between the immune cell or some kind of APC process, then I think you might look at a different technology because the membrane shape would be very important. But if you just want to know something like relative survivability under treatment, then I think even simple fluorescence-based counting should be sufficient.
A comment is that it seems like PDXNet has made a lot of progress. What are the next steps, future directions for the network?
Well, I think the number one thing that that's important for the field is to speed up the clinical trials. We want to make that go faster. Certainly having the increase in models will help us do that and choose the systems to study. But we want to improve that timeline. And I think that we will get that partially by just picking the models quickly. But I think that this space where we're doing combination of organoid and xenograft, we need a lot of thought in how to do that very well. Because a PDX study takes, again, if it goes well, could still take a year. And it would be much better if we could reduce that to, say, 6 months. So I think that's the key question.
Funda, one more drifted into the chat box. It's probably our last question.
Are tissue arrays linked to these models available for collaborative biomarker screening?
I don't know, actually. You know, PDMR does things like that, but I don't know if they specifically provide that service. I imagine there's a way to generate that, but I don't know the logistics.
I can share that we have over 200 and something PDXs on TMAs across 30 histologies. So always happy to collaborate. Okay, looks like that's a wrap. Thank you, Jeff.
Okay, thanks a lot, Funda, and all the organizers and the questioners as well.
So I too would like to thank Dr. Trang for an excellent talk and Dr. Merrick Bernstam for excellent moderation. And remind everyone to please join us on July 28th. Our next talk will be by Dr. Fedorov, who's going to talk about the NCI Imaging Data Commons, part of the Cancer Research Data Commons. Thanks, everyone. Appreciate it.
Thank you.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.